Yeah. This one.
Mwalimu ajiingia ndio maana nasema
and 697
wakipiga makelele wakingojea amka.
Come siete kunira rekodi? Nijeje maundu mahana atia no ndamenyire. No ndamenyire ndarenda guthima ciki.
The Bluetooth device is the medium aux input mode. The Bluetooth
Okay, good morning, students. Victor Morara, are you getting me? Raise up your hand, Victor, if you are getting me. Victor, Duncan, Arene. I can see, that's very nice. That's very okay, I can see you. So, okay, we can start off. Last time we were discussing about cell division, and I even gave the question on cell division. Today, I want us to continue with the cell division. And in this case, we are basically going to look at, we say that before any growth occurs, just after reproduction, cell division must always start. So we say that we must first of all look at cell division, and we say that there are two types of cell division. That is mitosis and meiosis. So before you look at reproduction in plants and reproduction in animals, first of all, we must look at what we call cell. I must confirm that people are getting me. Cephas Mutua. Cephas Mutua. You are raising up your hand. Cephas. Cephas, you have a problem? Njoroge Stephen. Once again.
I'm good.
So we can go on with where we stopped. We were talking about cell division, and we said that the cells normally divide, and as they divide, they multiply and replicate so that we have so many cells that are formed. And when we finish this topic of reproduction, we shall go back to look at what we call growth and development. So we had introduced cell division. We looked at the structure of the chromosome, and we were able to say that the chromosome is actually made up of the parallel strands. This is just reminding ourselves. Abel, raise up your hand if you are getting me. Abel, Charo, nice, very good, thank you. So we say, we first of all look at the structure of the chromosome, then we can move on to look at each stage of cell division. We had said that we have what you call haploid and diploid. Na tukasema ya kwamba in haploid, it is where the number of chromosomes are half the parent cell. But when it comes to diploid, it is where the number of chromosomes are the same as that one of the parent cell. So we discussed about mitosis. I remember we were able to look at the first one, interphase there, and we say that during the interphase, the cell is only preparing to divide. Therefore, we cannot be able to look at what is normally happening. We cannot be able to look at the structure of the chromosomes to identify the stage of cell division, but instead we can be able to look at we need to understand that for the cell to divide, what must happen. And we say we have what you call up here, maybe if you may see where we have this multiplication, we say we have duplication of genetic material, so that the number of chromosomes that are going to be formed is either have the same number of the chromosomes as the parent cell, or it can be the same number of the parent cell. Then synthesis of new cell organelles, somebody had mentioned about the roles of the ribosomes, the roles of the centrioles, and even mitochondria. Cell division requires energy, and therefore for the cell to divide, it requires energy. Synthesis of energy, that is by mitochondria. And then we talk about that the chromosomes are not visible. We cannot be able to use the appearance of the chromosomes. Straight away, we look at prophase. That when you look at these structures, on each one of them, there must be one distinguishing feature. Look at these chromosomes during the prophase. Upper cut. The major feature that we can be able to see here is that you can be able to note that the chromosomes are thickened. The chromosomes are shortened. Zinaweza onekana as thick structures, thick threads. Then, when you look at these chromosomes here, so like this one here, the centrioles move at the opposite, the chromosomes thicken and become visible. So this is the major point here. And the centrioles move at the opposite poles. Opposite poles kumaanisha ni huku na huku chini. At the opposite poles. These are the centrioles. Last time we cannot say that centrioles take part in cell division. The Form 1 can say that, but a serious man, a Form 3 man like you next year, you can say they take. It is responsible for formation of spindle fibers. The role of centrioles is that formation of spindle fibers. Don't write take part in cell division like a Form 1. Then we were able to, from after the chromosomes can be seen, can thicken, the major distinguishing feature of metaphase is that they align themselves at the equator of the spindle fibers. Well, I say it's very easy to distinguish between prophase and metaphase. Metaphase, middle, look at how the chromosomes are. Kwa mtihani unapata hiyo structure, the chromosomes are at the equator, or at the center of the spindle fibers, and one is asked to identify the type of cell division, not stage. Type of the cell division, ukiona the chromosomes are not in pairs, this is what we call mitosis. Stage of cell division, it is metaphase. Kama zingekuwa, like if we just look at this one, hiyo ni metaphase. So the type of cell division, I'll say mitosis. Able to meiosis 1. Look at that. If you can be able to see meiosis 1. And meiosis 1, when you talk about metaphase, the homologous chromosomes happen. The chromosomes are in pairs, like it is still at the equator of the spindle fiber. So if I was asked to give this type of cell division, that one is basically meiosis. And then the one that is this stage is metaphase 1. I was only trying to bring to you so that you get the difference between metaphase 1 and metaphase. Are you able to distinguish between if I were asked the stage and if I'm asked the type of the cell division? So you go back to up here. So we are looking at what you call the metaphase, major distinguishing feature, and you see the centrioles migrate to the opposite end of the spindle fibers. This is not the major distinguishing feature, but the major distinguishing feature here, it is the spindle fibers. I'm looking for these ones. The chromosomes migrate and align themselves towards the equator fibers. The same as spindle fibers we have. This is what you call the fibers. From metaphase, and then we have anaphase. What happens in anaphase? Tulisema ya kwanza in prophase zinakuwa katika, then zinaachiliana hivyo. So they break at the anaphase. So at the anaphase, what happens, you can see daughter chromosomes. Now, wakisema daughter chromosomes wanamaanisha the outcoming chromosomes. Daughter cells are the cells that are resulting from them. So we can move on, and then you look at the major distinguishing feature in anaphase. We don't say chromosomes, but we say chromatids. Chromatids separate. So and they move on the opposite poles. Opposite poles zina separate hivyo. Alafu finally zina rejoin. So when I see them, known as chromatids, I'm told to identify the type of cell division. It's mitosis division. Kama ni half ni anaphase. Anaphase. I want you to look at anaphase here, and then we look at anaphase 1. So what if I'm told when is the answer expected to be anaphase 1? Or let to look at anaphase 1. Look at anaphase 1. Hii na hii zimeshikana. So this and this and this. So if I'm asked the stage, unaona zinakaa kama the chromosomes. These are chromatids that are in pairs. So if I'm asked to give the stage, then I'll talk about meiosis. The type of cell division meiosis. Na kama ni type of the stage, then it's anaphase 1. Watu angalie anaphase 1, and then we have to look at anaphase itself. Anaphase, it is very different. Happen in pairs. So here the other one is in. By the way, to break these chromatids, to separate them. So the first one here, the spindle fiber shorten, pulling the chromatids towards the opposite poles. Alafu, the movement of sister chromatid begin at the center, forming a V-shaped. Hii ni kusema to look at the term sister chromatids. Reason for your answer, and you say chromosomes separate with the sister chromatids. You unaweza sema two chromatids which move to the opposite poles. And I'm very happy with your talk from Chris. Away from this one, please. You have to be responsible men at home. So do that, because next year we need you men here in school. So make sure you take your time. Usikuwe tu mtu wa kuzunguka, please. Sometimes it's important to get the advice. Just if your parents are maybe you stay in your own home, that's the best. If you stay still on these rental houses where you have are they called whatever, your watoto wako wengi, minimize the movements, and it's for your own good. You don't do it for your parents to see. You don't do it that wazazi waone huendi. It is the right thing to do. Otherwise, tukikaa tu hivyo, you just move and then you feel like your friends in other schools. Sometimes we walk out here and we see students from other schools. Na natumia the good term, other schools. Watoto wamejaa all over, girls and boys all over, moving from one place to another. Unaweza shangaa kama mtu wa Mang'u kama wewe unaweza kuwa hapo, kwa sababu hata fikira yako ni tofauti bwana. Have you ever noted that hata in the future watu hawa schools wana move there, they're expecting jamaa wa Mang'u watakuwa na lead country, because your level of thinking is just different. Your level of thinking is different. Si wana expect jamaa wanakuheshimu. Hata unaweza pita hapo na ambia jamaa, man, you are risking so much, because they expect the future leaders are you people who reason out well. And I'm happy to see all of you. You can see Raphael there. Abel, I can see you. Owen Murik, Owen should be Collins. I can see you. That is very important, very good. Austin, I can also see Benjamin. Bethuel, you are from Trikani, but I'm very happy to see you, man. Kitu mzuri ya bahati ni kwamba next year bado tuko form 3. Thank God. Tuendelea vizuri sana. So we can move on. So that was just by the way, that make sure you behave. A Mang'u student is different from any other student. The way you think is different from any other student. Now, usijaribu kujicompare now. Don't see that your friends are going to play and you say, let me when tofauti tu. You are a unique man. Play with yourself there, do your mazoezi there, and avoid by all means. That was by the way. Telophase. Tumesema what happens to the first one. The cell it prepares to divide. Interface. We know the 3 characteristics that or activities that are taking place for this cell to divide. 2, after now the cell has prepared, the chromosomes align themselves at the equator, and finally, the chromosomes are going to separate. But now when they separate, we don't use the term chromosomes, instead tunatumia term chromatides. Then we talk about telophase. They rejoin again. Huko mwisho they rejoin, but I want you to look at the behavior. I want you to look at the behavior of this cell towards the end. What is mitosis? Type of cell division in which the cell divides to form 2 daughter cells. 2 daughter cells. Each daughter cell has half number of the chromosomes as the parent cell. Each daughter cell has half number of chromosomes as the parent cell. Hiyo ni kusema ni that when I wanted to get something to display it, but when you look at it at the end of it, it's almost dividing. So it's very easy to distinguish between telophase. But now, what about telophase 1? Remember, in mitosis, the cell will divide to form 2. So tunatarajia kwamba these 2 daughter cells are going to be formed. Lakini mwanafunzi anaweza distinguish aje telophase na telophase 1. In telophase 1, remember, the cell is supposed to divide to form how many daughter cells? 4 of them, 4 daughter cells to be formed. So hii ni tofauti because when you look at this one, 2 daughter cells are going to be formed. But when you look at telophase 1, look at this. Look at these ones. Here, it's ready to divide. But the distinguishing feature, upper chromosomes are in pairs. Alafu turudi hapa. It is just 1 you can see in chromosome moja, hiyo ni mbili, the two chromatids rejoin together. Look at the first one. During this, the chromatids arrive at the pole position, and then cell wall extends inwards, leading to separation of the cytoplasm, and that division of the cytoplasm is known as cytogamy. Cytology is the study of the cell, but when the cytoplasm divide, not the what? The nucleus. Nucleus divide inaitwa karyogamy, but when the cell divide, cytoplasm dividing, this one, it is called cytogamy. So there's separation of the cytoplasm, cytogamy. The process which cell wall grows inwards, leading to formation of two daughter cells. The distinguishing feature is that the chromatids rejoin at the opposite poles. Then, so this is trained, at least man, you have each characteristics. Hata wakati unasoma, na nilikuwa naambia from 3. Now you have good time to revise. Hata wakati unasoma, usiamke tu because you want to prepare for biology na anza kusoma. Sema tu leo, I want to look at mitosis alone. Cell division, importance of mitosis, how does it occur? That is the role, importance of mitosis, and the difference between mitosis and meiosis. In our chair, I go to play. You have good time to prepare and be a medicine student, as you have always dreamt of. Look at this one. I display this one because students will always hate it. But it's not. It's nachanganya. They don't. Look at this one. Wakati naangalia hii, we cannot be able to see the chromosomes, but we can see. We know that here during the interphase, it's preparing to divide. Hata nikiulizwa, state the characteristics, the activities that are taking place. Hapo, hapa, you can see they can be seen. Alafu hapa, prometaphase zinakaribia, but now here, ziko at the metaphase, ziko katikati, ziko half, zina rejoin. Nilikuwa naambia wale wana southwest na southeast that you can even draw five circles. Tuna omit gani hii because this one we don't see the chromosomes, but tunaweza anzia hapa so that they can be seen, they align themselves, they rejoin. So wakati unachora hizo tano, wakati unapewa kama niko mtihani, unaona tu hizi ziko half anaphase, but is it anaphase 1 or is it anaphase? Anaphase 1, is it anaphase? Is it metaphase 1? Is it metaphase? Then after looking at this stage of cell division, we can now look at significance, importance of cell division. And which cell division are we talking about? Mitosis. Let's have some discussion. What do you think is now the importance of cell division? You raise up your hand and unmute you so that you talk. What do you think is the importance of cell division? Raise up your hands so I can be able to see you. And I'm happy with your turnout. I can see over 200 people. That is very, very encouraging from 3s. What's the importance of this cell division? Man, tuongee tu bwana. Hapa hakuna uko tu kwenu tu na umetulia tu. We are just thinking is the importance of cell division. I'm waiting for somebody to raise up your hand so that I can unmute you. Raphael, you look like you can say anything anytime. When you raise up your hand, I'll be able to see you there so that you move on. I can see Raphael is there.
It leads to retention of good characteristics from the parent cell.
Yes, come up again.
It leads to retention of good characteristics from the parent cell.
What that means, very true. Anasema it leads to retention of good characteristics from the parents. Tusitumie the term good characteristics, but retention of the genetic material. Si tumesema in mitosis, the chromosomes that are being formed are the same as those ones of the parent cell. Diploid condition. So it's true that it can lead to retention of the characteristic from the parent cell. Very right? Any other man? Raise up your hand so I can be able to see you. This is lesani. Kidogo tu lesani. Tunaji.
It is the basis of sexual reproduction.
It is?
It is the basis of asexual reproduction.
It is the basis of asexual reproduction. Remember, mwanzo tulisema, very true, tulisema that mitosis normally takes place in body cells, the somatic cells that is. So that's to say, if it takes place in the somatic cells, then that's where reproduction, basis of asexual reproduction. Let's have the last person. Najifunza tu pia mimi kujaribu kupata watu kwa mute na so na take time you understand hali ya. I wish I was in a school like Mang'u, like you now, because we do understand you easily to you. I'm waiting for somebody to raise up the hand. Somebody else? The last person, then we can move on. We can go on. We can go on. We can go on to save on time. Significance there. One of them, Rafa has told us that it leads to retention of the same number of chromosomes as their chromosomes therefore maintaining similar characteristics within a particular species. Here ni kusema tu, you don't need to use all these terms. In mind, you know, in mitosis, cell division in which the cell divides to form two daughter cells, each having the same number of chromosomes as the parent cell. Na ndio tunasema ya kwamba, if it's that way, then we can say that the daughter cells formed have the same number of chromosomes as the parent cell. It's retaining the characteristic. It leads to multiplication. To me the term multiplication, to me the term, ile lesani ametuambia, it leads to multiplication of unicellular organisms, each bacteria and amoeba. Munakumbuka bacteria, amoeba, they undergo what you call cell division, reproduction, but which type of reproduction? Asexual reproduction. So like bacteria, amoeba, these ones that will split into two, what we call binary division. So in short, hii lesana ametuambia that multiplication hii ni kusema it's the basis of what we call asexual reproduction. It leads to growth due to increase in the number of cells within an organism. Since it takes place in the body cells, then it can lead to growth. Because meiosis, as we shall be looking at significance, it leads to formation of the gametes. So this one is taking place in the body, it leads to formation, it's the basis of growth. And finally, it leads to regeneration of body parts of some organisms, each lizard. Nasikia lizard, normally the tail is fat, but it must have, because it is that part of the body. It takes place in the body cell, then it leads to that. Meiosis, I want to allow somebody to read, to take you through as I get something out. Some so that you come on the Abel Taro. Abel, raise up your hand. Get up so that I unmute, I can see. Nimekuona a minute kidogo tu. Abe?
So meiosis. So meiosis is a type of.
Before you reach there, you try to talk about it as I come. And then
Yeah. So meiosis, this is a type of cell division that occurs in reproduction in the gonad cell. So when you talk of meiosis here, the cell divides to form 4 daughter cells, each having the half number of chromosome as of the parent cell. So here, if you talk of meiosis, the cell divides to form 4 daughter cells, each having half number of chromosome as the parent cell, what you call haploid. Now, in meiosis, it normally occurs in the gametes, and therefore, it is responsible for sexual reproduction. Now, if you talk of meiosis, it forms or it takes place in two stages. So it takes place in 1, we have meiosis 1, and 2, we have meiosis 2. So there's 2 stages of formation of meiosis. So each phase consists of 5 stages. Now, if we start with meiosis 1, we have 5 stages, which are the same as mitosis, but here we only add, like, if we talk of interphase, we talk of interphase 1, we talk of prophase 1, metaphase 1, anaphase 1, and telophase 1. Now, if we talk of meiosis 1, here, we go to interphase 1. Now, in interphase 1, during this stage of meiosis, the cell prepares to divide, and we have the same activity as that happened in interphase. So we have the replication of genetic material, formation of new cellular organelles, and thus we have the buildup of energy necessary to drive meiosis activities. So interphase 1 is just the same as interphase. And then we go to prophase 1. Now, in prophase 1, the only difference between prophase and prophase 1 is that if you talk of prophase 1, you talk of homologous chromosome, whereby 2 chromosomes are joined together. So whenever we talk of prophase 1, whenever we talk of prophase 1, it's just the same as prophase, but here we add the term homologous chromosome. So we go to prophase 1. Now in prophase 1, just as in prophase here, but in addition of homologous chromosome. So in prophase 1, we talk of the homologous chromosome associate with each other. I mean, there are two chromosome joining. And also we talk of the homologous chromosome also the thicken and short, yeah.
So thank you so much. Thank you so much. I can pick up. Thank you for that. That reminds me that Mami Sana Shule. I can see when Mohammed Farah is there. Mami Sana, you people have been, Mang'u students who are mnafanya hata sisi tunachanuka so many things. Sometimes you get teachers relying so much on students because there's so much to learn from you in terms of behavior. See, sometimes when we hata kwa class naita some of you to assist us in connecting the projector because there is so much we learn from you. You're about to assume that all of them well. Meiosis, I hope you have discussed that it's normally occurring in the gonads. Gonads, these are reproductive. There are 2 types of gonads. You have the male gonads and the female. Human cytokines in the ovary for female and in testes normally in female. That one I know you have discussed. Sasa tofauti nimekuja nimepata tu in Abel was trying to discuss about distinguish between kama tuanze na interface. During that interface, it's the same during the interface 1, interface 2, and even interface. The activities are the same because the same is then when it comes to say process, you now need to discuss process 1, process 2. Na sasa wakati tulikuwa tuna discuss mitosis, tulikuwa tuna distinguish between mitosis 1 and mitosis. That's 1 and mitosis 2, so that we get the difference. But however, majorly in exam, you majorly look at mitosis. Mitosis. Metaphase and metaphase 1. Metaphase 2 is not likely to be asked because here it's like metaphase up there. It's only that the cells are now in 2. Kama uko na swali, you just raise up your hand. If you have any question, kuna kitu sijataja, na unajua simesema sasa hivi you are at home to kwa freestyle. You just raise up your hand, you will come on the screen there. I'll be able to see you and then we ask. If you have something, studio to hapo. Then we can go on. I just want to confirm that people are there. Donna, I can see you. John Nganga, just want to see Mwanzia. Peter is there. Peter Gitawa, I can see you. Just want to confirm that people are there first. That is right. So we can go on. Isaac Hamisa, I can see you. Sam, Nakuona, I thank God. Jamana ito Collins Warui pia. I can see all of you, very good. So when it comes to interface, it is the same. Here at red light, let's not talk about it, but implication and build-up of energy the same. What process? During this process, nucleolus and nuclear membrane disappear. The chromosome pair up. In a process called crossing over, the centrioles migrate. Look at this. During this, there is exchange of genetic material. This is NB. The point of contact between the chromatids is referred to as chiasmata, synapses and chiasmata. So the synapses is that line side by side. When they lie side by side, like this one, under hii color gani, this is maroon maybe, and the red. Hapa katikati, they can lie side by side, but the point of contact penye zimeshikania is what we call the chiasmata. This is another unique activity here. One stage there is during this stage there is exchange of genetic material. Very important stage. To answer genetics from 4, still we shall have to look at it. We shall have to refer to this one because then by the stage where we have cell division, where we have exchange of genetic materials, it is during process 1. Crossing over is during process 1. That can improve the quality of the species. It is during process 1 when we have exchange of genetic material. Metaphase 1, distinguishing feature, we say the spindle fibers are attached. Sorry. Nilikuwa nasema tuliangalia metaphase, prophase na prophase 1. Tunaweza distinguish. Na nataka tufungue prophase 1 na prophase 2. We'll be able to see the difference. This is prophase 1. Look at it. Look at chromosomes pair up. But look at it. Unajua in the first point is ninaweza sema here it is only to form 2 daughter cells. The 2 the two daughter cells that have been formed are also dividing. So look here. If it was prophase 2, another cell will be here. Kwa sababu during prophase 1, the first parent cell has divided to form 2 daughter cells. So tunaweza kuja hapa kama ingekuwa prophase 2, another cell will be here with the same characteristics. Look at it. Iko hapi. Interface. Look at prophase 2. Ni rahisi sana to distinguish between, you can see they are in pairs. Huh? There are 2 cells here. Where are we getting 2 cells? Because in prophase 1, the cell has divided to form 2 daughter cells. In prophase 2, in a part, the 2 daughter cells to divide to result to 4 daughter cells, each have half number of chromosomes as a parent. Wanafunzi yako tu very careful. Don't need to read it in this. Prophase, kwa hivyo tunaweza distinguish between prophase 1 2. In prophase 1, they are thickened and be seen alone. In prophase 2, the chromosomes are in pairs but still thickened. In prophase 2, the chromosomes are still thickened but the cells are 2. So that at the end of it all, unaona itakuja metaphase bado stili telophase mbili, and finally when it comes to the anaphase, you can see there are 2, but now finally when it comes to telophase they divide. They divide. You can say from 1, 2, 3, and 4 to give us 4 daughter cells that have the half number of chromosomes as a parent cell. So when we look at metaphase, you can see at the equator of the spindle fibers, again, saying it is metaphase 1 and not metaphase, it's different. When it's metaphase, and the reason for your answer is that the chromosomes are at the equator. But when it comes to metaphase 1, tunatumia the term homologous chromosomes are at the equator of the spindle fibers. Man, they are in pairs, man, you can see, but at the equator of the spindle fibers. Telophase the same, you can be able to see them. Interphase 2, they repeat. I don't need to repeat this one because ni kama zile za kwanza. It repeating the same build up of energy for cell division and the same. Telophase 2, you can only see the difference is that now they are in 2 cells, not 2 chromosomes, 2 cells that will finally give us 4 daughter cells formed. Significance of meiosis leads to formation of gametes. Somebody said because it's occurring in gonads, then it can lead to formation of gametes. In male, it can lead to formation of sperm. In female, it can lead to formation of the ova. Leads to variation in species. In species. How? Unajua during that cell division, the cells normally, and during which stage? Sorry, kidogo. Fine. Yes. Kumekuwa na kitu kidogo, sijui ni nini, net imekwama kidogo, but the chase and mathematics. I've just checked if she's there, but kuna kitu on internet. Wacha nimalize kwa free next. Then check on the internet. Niko kwa nini tunaweza kuja tu? Sawa. We can go on. It leads to variation. Specifically at what point? Then it is during that stage of prophase. Now, tumalize na hivi. We look at the differences similarities between mitosis and meiosis. We have looked at all these. Let's have similarities and differences. You can raise up your hands now. Raising up your hands, what you do, there's something there, reaction, you click it, and then I can be able to see. As I know, you click it. You do that physical, similarities. Similarities. Ewa se tuongee bwana. Hata Bruce. Austin Kimenjo akiendelea kujipanga na yake. Bruce. Oh, I can see people are raising up the hands. Vitu bwana najifunza polepole. Now where are they? Steve Mwangi, you can see you. Shida ni sasa kumute hawa jamaa. Steve Mwangi, you are there?
Mitosis and meiosis both involve duplication of chromosomes.
It is?
Both involve duplication of chromosomes.
Very good, very good. That in both the genetic material must replicate. Because hata kama ni chromosome itakuwa half, but 4 of them in meiosis, 4 of them having half, it must duplicate. Very true. Ingine? Farah ngoja kidogo. Bani hizi vitu za kumute na panmute. Farah? Jesse, you're raising up your hand. Limo.
Both take place in plant and animal cells.
Both are going to take place in plants and animals, very true. Lesani, you are raising up your hand. Kidogo tu Lesani. Najifunza polepole, mnanipeleka polepole.
Both involve division of cells.
In both, there must be division of the cells. Let's look at differences. Let's look at differences, then we can go to, I want 5 people to raise up your hands. Farah, you can raise up your hand before. Victor Kigo pia, acha kwanza tungoje Farah tuone. Farah. Sorry, Farah ngoja kidogo. You are still in the meeting. Abe?
Yeah.
We can get you. Victor. We are looking at differences. Nataka tupate tano, I want 5 people, then we move on.
Difference.
Man, we are looking at difference between mitosis and meiosis. After we've discussed what is the differences. Kuna huyu jamaa anaitwa nani, Sami Mushiri, nijaribu kuone kama hata. Sami Mushiri.
In mitosis, it involves asexual reproduction, while in meiosis, it involves sexual reproduction.
Very beautiful. Benjamin Kinudia. Zakaria, what you do? Click for reaction. Acha Benjamin atuambie kwanza.
Mitosis occurs in somatic body cells and meiosis occurs in gamete cells.
Very smart. Ya mwingine anaitwa Bruce Gidaiga. Bruce. Kidogo tu Bruce. Man, hizi vitu. Bruce, a minute.
In mitosis, it results in 2 daughter cells, while in meiosis, there are 4 gametes which are formed.
Very smart. Mohammed?
To reduce gas.
Then ukishamaliza kuongea this man called Edwin Waguru. Abel?
In mitosis, the process is shorter because it involves only one stage, while in meiosis, the process is longer because it involves 2 stages.
So in short, you are trying to say that mitosis takes place in one successive cell division, but meiosis takes place in 2 successive cell divisions. Sindio?
Yeah.
If somebody has something, somebody is Edwin, speak up. Victor, sorry, Victor Kihigama.
In mitosis, the daughter cells are genetically identical, while in meiosis, the daughter cells are different from the parent cell.
Because unajua kwa nini inakuwa hivyo, Victor? It's because in mitosis it is from the body cells. So the body cell inaweza toa tu direct ifanane. That's why you are saying it can be identical. But in meiosis, because it involves fusion of genes, then it can result to, depending on which gene is strong, which gene is dominant, then the resulting offspring may have a slightly different characteristic as from that one of the parent cell. Thank you very much. You can see people are raising up the hands. I want us to move on. Na in the next lesson, nataka nitatoa paper, nimepewa some good paper. We shall discuss. Hatutaendelea na lesson next week, tutaendelea na lesson, we shall discuss a paper. I'll display it there. Mtakuwa nachagua one person anasoma swali na jaribu to explain, and we discuss the paper. So, and today, I'm very happy that you people have not done it on my screen like it has been. Tukishamaliza hapo, we shall start on sexual reproduction. Na nafikiri tutakata I finish up with the sexual so that tukimaliza ku revise hiyo paper hiyo ingine, any other time we can go to sexual reproduction. Kwa sababu hii ni mention tu. So you just give me a few minutes. When it comes to asexual reproduction, types of reproduction, we have asexual and sexual reproduction. I'm finishing in the next 5 minutes. So this organism developing, forming a new and free living organism. And in this case, they are of 3 types. We have binary vision, and binary vision it normally occurs in the amoeba, like that one of bacteria. Spore formation here, rhizophus and in yeast. So we look at how does binary vision occur, splitting into 2. How does spore, how does it occur? Kwa hii, you need to look at how the spore is normally formed. And on this budding, we need to look at how the sprout is able to be formed on the cell that is able to develop. Binary vision. by which a single cell organism, e.g. amoeba, is there, splits into two, giving rise to two free living organisms. I think I'll do this one. We have this KCSE paper question that is this one, and that one for the spore formation. I displayed there so that we look at where. Then in the first one that the cell will always divide into to form daughter cells. Begins by immature cells synthesizing new cell organelles. That is at what point? Interphase. In asymmetry, it begins with the new cell organelles being formed. The cell organelles are taken division. So kitu ya kwanza you are telling the examiner, if I was discussing binary division, that it involves split and then begins with of new cell organelles, just like it is in interphase. And then followed by nucleus dividing into two equal parts itself. An outgrowth in the cell which splits the cytoplasm to form two identical cells. Jamaa mwingine ametuambia hapa at mitosis, the twins that have been formed are identical, the resulting offsprings. And it's actually that type of mitosis. Na tukasema ya kwamba the splitting of this cytoplasm is referred to as cytogamy, while this one of the nucleus is referred to as karyogamy, K-A-R, karyogamy. The new cell freely exists, matures, and divides in the same process. Sasa, watu, can you be able to sema kwa mtihani ukiuliza to briefly describe how binary division occurs? Because you are a science student, can you be able to use your own words to explain? Where's the kosa ya kwanza we are talking about? Look at this. Ndio nilikuwa nasema wakati unasoma just try to go for understanding. The first one we are trying to say that one, the first thing is to look at cell division. Two, interphase. You tell the examiner that it normally splits into two. Two, it's followed by the interphase, formation of new cell organelles, division of the nucleus, division of the cytoplasm, formation of the sprout on it, of the new cell organelles existing freely. And it's there on its own. And you look at this one. Hapa wameanzisha, there it is, and then we move on. Spore formation, also referred to as sporulation. Some organisms, each bacteria and fungi, that one you can be able to see. But of the most important, look at that structure, not budding. Allow me to stop on spore formation, please. Budding is there. Sorry, we have spore formation there. We have it this one. We have it here. Spore formation. Uye ni nani? Ronald, you are raising up your hand. You have something? You have something, Ronald? Do this so that I know you have something. Okay. Spore formation. It is here. Spores are here. These are the spores. This is the sporangium. The tunda in here, it's the sporangium. And the sporangium, so these are the vertical. The horizontal are the stolon, and we have the rhizoid on that. So you can also still be able to describe that one. For our time, we have discussed much. I'll request that we stop there. Thank God that today people have been very much cooperative. The next lesson umesema itakuja na paper ya form 3 imefanya somewhere, we discuss. Then ile ingine we can go on with the revision of our topic today. So at your own pleasure, you can leave the you join in another lesson. Thank you so much. God bless you. We need you next year at home. Maana tumemaliza class, watu watoke. Kama hujui kutoka unaita dada yako mdogo akuja kutolee kama hujui kutoka. And I can easily see wale wenye hawajui kutoka there. Tafuta dada yako ama ndugu yako mdogo mwenye amesoma kompyuta akutoe. Steve, kumbe hujui kutoka bado huko bwana. Hata Ronald ameshindwa. Hata Collins Warui. Unajifanyanga unajua kompyuta, hata Sean. Ita dada yako akutoe. Collins Mwangi. Lesson imeisha. Mwanzia Peter, let me call your brother akutoe. Some available, I can also call your brother. Acha nipigie mama yako akuje kusaidia kutoka. Au naona unaweza shinda hapo the whole day. Tutoke wote. Kuja sasa niko kwa kuja tu nimalize lesson. Just come. Okay.